Well, good morning, and thank you everyone for being here. Let me just get my presentation started. So can you all see the slides? Can you see the slides? Yeah. Okay, well, good morning, everyone. The work I'm going to talk to you about this morning is made possible by a very collaborative and scientific interaction between two institutes called the Tata Institutes for Genetics and Society, otherwise called TIGS for short. There's one at the University of California, San Diego, and the other one exists in Bangalore, where we have been in operation for about a little over a year and a half at this point in time. Now, it is my pleasure as the global director of TIGS to be a co-sponsor of this first ever India Science Festival. I've been passionate myself about science, and it is wonderful to see young minds get interested in science and ask interesting questions. And hopefully you can turn this, you don't have to be a scientist to be interested in science. You can do this in your daily lives, as you heard in the previous session. And it's good for the public to be educated in science, because so much of what we do today and what's happening around us is, in terms of excitement and new discoveries, is happening in the broad area of science. And this is science is what translates into technology. Now, I intentionally chose the title as a teaser because how many of you know what the world's deadliest killer is? Any hands? Did you look on the internet? Well, at least you're curious enough to show up. A few years ago, Bill Gates called the mosquito the world's deadliest killer. And he was slightly wrong because he wanted to make a point, because the mosquito is not really the killer. A mosquito, when it bites you, will not kill you, but it has the ability to carry pathogens like viruses and parasites, which are transmitted through the mosquito to the humans, so that you get diseases like malaria, dengue, chikungunya, Zika, yellow fever, and so on. So I've changed the title to say the mosquito is the world's deadliest messenger, not necessarily the world's deadliest killer. And the question that I'm going to pose to you, if you pay attention, is that should we shoot the messenger, kill off the mosquito, or should we actually disarm it by preventing its ability to transmit disease from one human being to the other? And that is a question that I hope, with a show of hands, you all will be able to answer. Now, all of this was made possible, the existence of TIGS was made possible by a very generous gift from the Tata Trust. And I want to tell you a little bit about Mr. Tata, Jamshedji Tata, who is the founder of this, because you're all looking for inspiring leaders and visionaries, and here was one that I want to tell you about a little bit today. Jamshedji Tata lived between 1839 and 1905, and at the age of 16, many of you are over 16, right? How many of you over 16 here? Most of you. He was already trading with China with his father, and so he set up some companies that worked and didn't work, and finally he succeeded in setting up a company called Empress Mills, which was making textiles in India and Nagpur and exporting this to UK and so on. And he was very, very appreciative of the fact that society was what enabled entrepreneurship to happen. And in those days, he set up something called the Tata Trust, and he was the first to set up the 8-hour work week, which world over we have 9 to 5, the 8 hours. He was the first one to do that. He was the first one to set up a creche for women with children. They would come to work. He set up a daycare center for them. He was the first to provide good working facilities, the first to provide pensions for individuals and gratuity and so on. All of the things that you take for granted today, he was the one who set it up in India for the first time. And he was a visionary, and one of the greatest things that I admire about him is this statement that is up on the slide, and that is that he said that what advances a nation or a community is not so much to prop up the weakest and the most helpless members of the society, but to really achieve this by providing help to the most gifted members so that they could be of greatest service to the nation. So he understood way before management schools and teams and so on, the concept of leverage, the concept of return on investment, and he decided that he would take the 2/3 of the shares of his company and all future Tata companies and put them in the form of a trust so that they could give back to society. And of course, the Tatas, you know, have been the first to set up airline industry, locomotive industry, steel industry, and you can go down the list. They were also the first to set up a variety of academic institutions in India, like the Indian Institute of Science, Tata Institute of Fundamental Research, National Center for Biological Sciences, and so on, Tata Memorial Hospital. So a real visionary who took what he earned and gave it back to society. So what is the mission of TIGS and why are we so excited about it? The mission of TIGS is that TIGS is a not-for-profit research institute that focuses on research training and capacity building, focusing on really new genetic technologies and trying to use these to alleviate food security and healthcare security in the context of India. So it is an institute in India with scientists of Indian origin working on problems of relevance to this particular country. And the areas of focus that we have, starting from the right of the slide, is first we want to focus on human hematopoietic stem cells. In your bloods, there are stem cells. These are the cells that give rise to all the different types of white blood cells and red blood cells and megakaryocytes and neutrophils and so on. And there are many diseases that affect cells of the blood. And these are in principle, if you understand what causes the disease, it's easy to cure them because you can do a blood transfusion or a bone marrow transplant, which is a standard of care at this point in time. So we've decided to use the technologies that I will talk about to try and understand how diseases are caused in the human blood system and how they might be cured. And this is simply a launching pad to study other diseases of relevance to humans. The second big problem that we're going to tackle, which is the second one from the right, is antimicrobial resistance. The world over, the antimicrobial resistance is a horrendous problem at this point in time, to the point where pharmaceutical companies have given up the search for new antibiotics because these microbes become resistant to these antibiotics pretty soon. And so if you end up with a disease that is multi-drug resistance, then there's not much hope for the patient in the hospital. So we are trying to reverse this process and try to understand it. The third area, which has to do with food security, is a focus on agriculture, particularly in rice. India has over 1,000 varieties of rice, and while you might have a rice with high yield or ability to grow under low water conditions, combining these traits into a single plant becomes prohibitively expensive, particularly when the number of traits are more than 2 or 3 or 4 or 5. And so we want to use these technologies to accelerate this. And the final area, which I will focus on for the rest of the talk, are diseases transmitted by the mosquito. The mosquito is the transmitter of the disease or the messenger, and therefore this is called a vector-borne disease, the mosquito being the vector. And there are many, many such diseases. So this is the area we'll focus on. And let me start with the disease malaria. So malaria is a problem that puts 3.5 billion people on the planet at risk for malaria. Any one of you can be bitten at this point in time, and including me, and I could end up with malaria if the mosquito has taken the blood from an individual who's been infected with malaria before me, and then goes through a certain cycle and then comes and bites one of us. So, and in every 2 minutes, a child dies of malaria. By the time I'm done with this talk, about 15 or 20 children around the world would be dead as a result of malaria. And in 2018, there were about 220 million cases of malaria, some 400,000 deaths. But the most important thing I want you to walk away with today is that every 2 minutes, a child dies of malaria, and would you like to do something about it? Right? And as dangerous as this sounds, other mosquito species, which are listed at the bottom, Malaria is transmitted by the mosquitoes of the genus Anopheles, but there are other mosquitoes like Aedes aegypti, Aedes albopictus, and so on, that cause a variety of diseases. So shown here are a variety of diseases at the top: Zika, dengue, chikungunya, yellow fever, West Nile virus, malaria, and so on. And collectively, these 5 species of mosquitoes shown here put 6.5 billion people on the planet at risk. And so this is an equal opportunity biter, the female mosquito. It is only the female that bites because she needs the blood to lay eggs and get proteins and other nutrients like iron from the blood. And so collectively, these mosquitoes put 6.5 billion people at risk on the planet, and it caused enormous devastation, and more people have died due to diseases caused by mosquito than by natural death through the entire history of the human race. So what is the relevance of malaria to the context in India? So it turns out that mosquitoes, as I've said, are the most dangerous messengers or killers on the planet, as Bill Gates dubbed them. But as I told you earlier, they are the messengers, not the killers themselves, and you should keep that in mind. Now, you might think that every mosquito transmits these diseases, but around the world there are 3,600 species of mosquitoes, and it is only a handful that transmit these diseases, and those handful are the ones that I showed you in the previous slide, and about 40 species under the genus Anopheles transmits malaria. In India, there are 4 or 5 such anophelines that transmit malaria. Malaria itself is transmitted by these anopheline species in India, and India and the world has declared that they will eradicate malaria by a certain time. India has picked the year 2030 to eradicate malaria. The WHO has decided that they will eradicate malaria by 2050. And there's, at this point, frankly, no hope to achieve these goals, because the existing technologies, which I will tell you about, are not working so well, and I'll come to that in just a couple of slides. The economic burden, the cost of tackling malaria is huge, because in 2017, about $3 billion was being spent around the world, and in 2020, we are already there now. That money needs to have doubled to $6.some billion, and so still there's no hope to get that kind of money. So we're not likely to be able to sustain this ability to reduce malaria very significantly without the resources that go behind it. And within India alone, it is estimated some years ago by the Times of India that if you take the total economic cost of malaria in India, it is about $2 billion US dollars in India alone. So what are the techniques and tools that we have to tackle malaria and this type of disease? We have a variety of tools at the present time. We have anti-parasitic drugs. These are the drugs that affect the parasite. There are 2 types of parasites, Plasmodium vivax and Plasmodium falciparum. These are the parasites that transmit malaria through the mosquito. These are not working so well because every time you create a new antiparasitic drug, the mosquito will become resistant to it in some period of time. Attempts have been made to immunize humans by vaccination, but these are not very successful because the mosquito again and the parasite keeps changing. There are also efforts to create insecticide embedded nets. So you can, as long as you sleep under one of these nets, you won't be bitten by the mosquito. But the mosquitoes are also evolving, so they have now evolved to bite during the day. So you can't sit inside a net all day and hope not to be bitten by a mosquito. So this is also creating some problems. Insecticides themselves are a bit of a sledgehammer approach, because when you spray insecticides, you're killing other insects too, and including breathing that yourself. And you may have seen, if you go in an airline, they'll come and spray some insecticide and tell you it's not harmful to you, but who knows? And also the mosquitoes and parasites are becoming resistant to these things. And then finally, you can reduce the amount of water that is there. All mosquitoes need is a little bit of water, but how can you dry up everything that you will, even a little lid of a bottle that you use for bottled water or for drinking water is sufficient for the mosquito to lay eggs and survive. And so all of these technologies, while we have had them and they have reduced malaria significantly, are not sufficient to eliminate them, which is why we need new tools. And some of these new tools are the tools of using genetics to try and tackle this technology, and some of these are called sterile insect technology, or genetically engineering mosquitoes to modify them to be able to not transmit the disease. So I want to take you back before I go into the details of this technology to about 40 years ago. I happened to be in the lab of the Nobel laureate who got the Nobel Prize for essentially creating some of the major tools in recombinant DNA technology. And you know that simplistically, recombinant DNA technology is the ability to cut segments of DNA from two different organisms and put them together and assemble them so that you now have segments of DNA from two or more organisms in one single organism. So shown on the left of the slide is the fact that you can create, as a result of this technology, transgenic or transgenic organisms which have, in addition to their own DNA, some foreign DNA in them. And so you can create transgenic plants, you can create transgenic mice, and so on. And shown on the right is the fact that you can create, using the same recombinant DNA technology, you can produce proteins of human interest. So if someone is short and they need growth hormone to grow, you can produce growth hormone and give them injections, and they will grow normally. And similarly, many therapeutics now in the industry are antibodies which are produced using recombinant DNA technology. So you have a variety of cancers. You will be given chemotherapy and radiation and so on, but also what are called biopharmaceuticals. And this biopharmaceutical industry has taken 40 years to develop and mature. And it was estimated that in 2016, the total value of goods produced by the biopharmaceutical industry is $192 billion. And by 2020, which is where right now, 2021, this will rise to $300 billion worldwide. And India plays a very significant role in this biopharmaceutical industry. Now, as exciting as this is, and let me use an analogy to what recombinant DNA technology allows, this is like a word processing software. If you had the ability in your word processing software to cut and delete and insert whole sentences, whole paragraphs, whole pages, but without the cursor to do this editing at the alphabet level within a word, this is what recombinant DNA technology allows. It allows you to move big chunks of DNA, just like big chunks of words from books and paragraphs and so on, to shuffle them around, and you can combine them from different sources to put it together. But you did not have the ability to place a cursor in DNA at precisely the location that you wanted. And this is a technology that was developed by scientists at MIT and Berkeley fairly simultaneously. And this is called the gene editing technology. And how many of you have heard the word CRISPR-Cas9? So many of you have heard this. These are the two molecular tools that are used. And CRISPR is simply a molecular cursor that, it is a ribonucleic acid which forms base pairs with the DNA, for those who have taken biology. And so you can place this cursor anywhere in the genome of any organism at a precise location. And then Cas9 acts like a pair of molecular scissors. It will come and cut the DNA, both strands, at that location based on where the cursor has been placed. So these two very simple tools, a molecular cursor and a molecular scissors, allow you to cut precisely a DNA of any organism at precisely the location that you want. Now, under ordinary circumstances, what happens is that such double-stranded breaks in the DNA are lethal to cells, all the way from bacteria to human cells, and the cells do everything possible to try and put these DNA segments together to join them, and this happens by a pathway called non-homologous end joining. It simply means the two ends are pieced together with a glue, which the DNA repair mechanisms exist in all of our cells, include from all the way from bacteria to plants to humans. And a fix is made, and it happens accurately, let's say 99% of the time, but if a small percentage of the time, a few errors are made, but it's better to be alive than dead because this is a lethal event if the ends could not come together. Now, that's not the pathway we're interested in. Instead, if you can take the same pathway and fix it in a different way using what's called HDR, homology-directed repair. I won't go into the details of that. It allows you not only to make precise edits, but also insert a gene of interest or GOI into that location, and this is the technology that I will use. So in other words, with CRISPR-Cas9, you can make a cut anywhere in the DNA of any organism, and you can put a gene of interest into that place using this technology, often using the enzymes and repair mechanisms that exist in cells. So this whole technology and the work I'm going to tell you was actually done by a PhD student. It's not, he was in a lab that had a great mentor and so on, but this is every one of you might be interested in going on to a PhD program. In other words, you could do this, and conceptually, it is very simple. So what did Valentino Gans, shown on the left, and Ethan Bear on the right, his mentor. Valentino Gans is a PhD student, and he was working with fruit flies, and he was working with fruit fly that is said to be heterozygous, and this is a diploid organism. It inherits one chromosome, one of those horizontal lines from its mom, the other one from its dad, and it is heterozygous because the two sequences are not identical. Shown at the bottom is the fact that he inserted a cassette with these molecular tools, the CRISPR and Cas9, he inserted it into the genome in a locus called the yellow locus of the fruit fly. This normally, when the yellow locus is functioning, it gives the cuticle or the skin of the fruit fly a certain dark color. And if you don't have it, then it becomes a yellow color. So because it is heterozygous and there's one copy still fine, this fly looks normal. And what he asked simply was that can he copy this cassette from the lower land, the CRISPR-Cas9, can this be copied with the molecular scissors and the Cas9 enzyme onto this chromosome on the top? And when he did this, indeed, it worked. And so here's CRISPR and Cas9. They go and find the molecular target. It now makes a cut there, and now that gene is disrupted, so now fly becomes yellow. So it has no functional yellow gene product, and so it now cannot make that colored cuticle, and it now has a yellow cuticle. And in the process, then you will notice that this segment of DNA has been copied from the bottom chromosome to the top chromosome, and now both maternal and paternal chromosomes have this DNA. Now, how many of you have had any introduction to genetics? Do you know who Gregor Mendel was? Yes, a few of you are showing your hands. So this was a Austrian sadhu, Austrian monk. He in his spare time was growing pea plants, and he discovered the rules of modern genetics 150 years ago. And the rules are very simple. So all diploid organisms, from flies to humans, we each inherit one chromosome from our mom and one chromosome from our dad. So if you intuitively say, what is, if only a mom or dad had a particular property, what is the probability that the offspring would inherit that particular trait? Is it 100% or is it 50% or something different? How many of you think it's 100%? Very few or none. How many of you think it's 50% chance of inheritance? And that is indeed the correct answer. I'm glad to see that many of you are suggesting it's 50%. And so this was the simple Mendelian laws say that if there's a set of chromosomes colored in different ways, one from mom and one from dad, showing you the fruit fly as an example, there's a 50% chance because one chromosome will come from each parent. And so shown here are 5 offsprings with one chromosome from mom, one chromosome from dad, which means then that the particular trait or property that we are looking at, it could be cuticle color, eye color, etc., is inherited 50% of the time. And this happens to be a trait called a recessive trait, meaning as long as one good copy is there, the trait will not be visible. So the 100% of the flies, in this case, appear to be normal because they don't have the visible trait, because there's at least one copy that is present of that particular chromosome. Now, let me take this to the CRISPR-Cas9 technology. So once again, we have two flies, one mutant, one normal, which is called wild type. Again, 50% of the time, they will inherit one of those chromosomes from mom or dad. And this is exactly like Mendel's laws as I showed you. But now, we have put in the cassette for the two molecular tools, CRISPR and Cas9, into the chromosome. So it has the ability to copy from one chromosome to the other, so that all the chromosomes from mom and dad have the desired gene edited. And therefore, what is the prediction now? Instead of having 50% inheritance, what will you have at this point in time with the offspring? Someone shout. 100%. Okay, very good. So you got 100% marks for that. And so that is indeed the case. And so now all of the progeny inherit the desired trait. And so the question came up, how can you use this technology for something useful? And this is where another brilliant scientist at the University of California, Irvine, came into the picture, and his name is Anthony James. And he had been working on malaria for quite some time, 20, 25 years, and he thought, how about the following? He knew that when a female mosquito bites a human being and takes the blood, that malarial parasite goes into the mosquito and it goes through the stages shown here. So what goes in are gametocytes, which are the male and female form of the parasite. These get converted into these things called oocysts or oocynids, and then eventually that goes from the gut to the hemolymph or the blood of the mosquito, and then transmits itself to the salivary glands, and when that mosquito goes and bites the next individual, the sporozoites are transmitted, and that's how the next person gets malaria. So he said, look, I can easily define 3 or 4 clear stages of the mosquito, of the malarial parasite in the mosquito. If I can block one or more of these stages, then I can use this technology to immunize the mosquito. Now, no one had thought at this point of immunizing the mosquito. I told you human vaccines don't work, but no one had thought, how can you block the stage in the mosquito? So he decided he would immunize the mosquito, and he was very clever. He actually said, let me take 2 antibodies from mice and take the genes for that, put it into the mosquito, and I will aim this against 2 different stages so that there's no possibility of getting resistance. So here's a simplistic experiment that he did. He took a mosquito chromosome and he inserted into it 4 genes, CRISPR, Cas9, and now 2 antibodies against the malaria parasite against 2 different stages. And this cassette is what I will call the gene edit. It is in one of the chromosomes. Now this mosquito will go and mate with another mosquito. And as you saw on the previous slide, when that mating happens, the gene edit, the 4 genes from mom get transferred to dad, and all the chromosomes, therefore, from both mom and dad have the gene edit, and therefore 100% of the progeny shown on the bottom line will have will become immune to malaria. So by using this gene editing technology, he was able to take a mosquito that is able to transmit the malaria parasite and make it immune to malaria. It cannot transmit malaria, even though this female can still bite. And the beauty of this technology is the following. This set of mosquitoes at the bottom row here become your ASHA workers. That is, they will go out, and as long as they can mate and find another mosquito, they will become immune and transmit the immunity and keep doing this forevermore. And the idea is that if you release 1% of the mosquitoes in an environment like this, within 10 generations or 1 year, from one monsoon to the next monsoon, 100% of the mosquitoes in this environment will become immune to the ability to transmit malaria. So this is the beauty of the technology. Mosquitoes are doing the work. You don't have to pay anything. You don't have to worry about unions. You don't have to worry about remote areas. Mosquitoes, based on the ability to want to mate with a partner of the opposite sex, will do the work for you. So this is what was done. And let me just illustrate this more graphically. So here's a human being on the left infected with malaria. Mosquito comes and bites. And that mosquito then goes and bites another human being after those multiple stages have taken place in the mosquito, right? And then now that human being gets malaria as a result. Now, let's take two strategies. So I come back to the question I raised in my first slide. Should we kill the mosquito and get rid of this messenger? Should we shoot and kill the messenger? And there is a technology to do that using very similar technology to what I just described. So what happens here is that if you use some technology to either change the sex ratio, or if you prevent females from developing or males from developing, very soon there are no mosquitoes to mate. And so the mosquito gets killed and disappears. And therefore, will the disease be transmitted now? So there are no mosquitoes. Effectively, it's the same as spraying or swatting a mosquito and killing it off. That mosquito cannot transmit disease. And so this is one strategy of what is called population suppression to get rid of malaria. Now, the alternative strategy, which is what we are focusing on because of a variety of advantages, is that now we have immunized that mosquito that first bites. And although that mosquito is able to go and bite just another human being, it has now been deprived of the ability to transmit malaria. So it cannot transmit malaria anymore. So this is called a population replacement strategy. And shown here are some of the positives and negatives of each of these technologies. So let's take the population suppression technology, for example. If you kill off the mosquito, the strength is that if that mosquito causes one disease or 100 diseases, all of those diseases will be wiped out. And this is important because there are mosquitoes like Aedes aegypti that cause dengue, chikungunya, Zika. So if you kill off all Aedes aegypti, then of course you would not have those diseases. And parenthetically, I will tell you that 75 years ago, India had no dengue, no chikungunya. Aedes aegypti is an invasive species in India that did not exist here. So if theoretically we were to wipe out Aedes aegypti, India would survive fine, despite the fact that environmentalists might tell you, oh, you're eliminating a species. But this is only one of 3,600 species of mosquitoes. Now, the disadvantages is that every time I kill off the mosquitoes in this room using whatever technology, then next year I have to do the same treatment again, because every year going forward, you have to keep doing the same treatment. And this is very important in terms of public health, because psychologically that last mile completion is the most difficult. India almost eliminated malaria in the early 60s, and then hardly anyone was getting malaria. You could hardly see very many mosquitoes. People said, well, there are more important diseases for us to work with. Money got siphoned to other places. And then 10 years later, it came back with a vengeance. And today, India is the number 4 highest prevalence of malaria in the world, and so it has come back with a big vengeance because we did not finish the job. On the other hand, the population replacement strategies that I just described have the strength that it is self-sustaining, and it also causes minimal environmental damage because the mosquitoes are not killed, they still continue to bite, but like the 99% of their brethren that don't carry the parasite, these mosquitoes don't transmit disease. They're a bit of a pain, but you can easily survive them. There's no big deal. The weakness here is that for each specific disease, you have to have a specific antibody or tool to neutralize it. But this is something that also gives you greater control, and so regulators might actually like that. So this then completes my talk. Let me just summarize here to tell you what I've just said. So I started with telling you that mosquitoes have been labeled as the world's deadliest killer, and I changed that to say because the mosquito itself does not transmit disease, it is really the world's deadliest messenger. And the question I pose to you is that should we get rid of the mosquito and shoot and kill the messenger, or should we alternatively use this new technologies that I've described, gene editing, gene drive, and new genetic technologies, to disarm the mosquito, to make it immune to the ability to transmit malaria. And if you can do this with malaria, you can certainly do this with dengue, chikungunya, and so on down the line. And so we have both of these technologies. And the question that I'm going to leave you with are the following, and I'm going to just read the questions out for you first. First, should we shoot or kill this world's deadliest messenger? The second option you have is option B, is that should we disarm this using the genetic technologies? The third is should we do both? And finally, D is should we do nothing at this point in time. So you're allowed to give only one answer, A, B, C, or D. How many of you think that we should have A as the answer, shoot and kill off the mosquito and get rid of it? So we have, let's say, 5 people on this side. How many of you think we should disarm the mosquito without killing it and prevent its ability? Perfect. So I've done a good job in convincing you that, and I'll just have a little joke about it. I presented this talk to the Indian community in San Diego, and one old man came and said, very good, you're practicing ahimsa. You're not killing the mosquito, you're actually preventing it from transmitting disease. So it also tells me, though, that you've paid attention, and so this is something for you to think about. How many of you think we might want to do both? Very good. There are, although this is a smaller number than those who want to disarm the mosquito, there's no wrong answer here, because there are cases, if you had a massive epidemic here, for example, you might want to bring down the population first as an immediate measure, and then kill off the mosquitoes using whatever strategy, and then stabilize it with the technology. And how many of you finally think we should do nothing about this? Brilliant. No one wants to do nothing. So each one of you can indeed do a fair amount. Start with preventing bodies of water around your home and allowing mosquitoes to breed, and then learn more about this technology through your classes and so on, because this technology that I've described to you is we've taken, let's say, the laws of gravity, these are laws of Mendel, which are the holy grail of genetics, and we've turned this upside down on its head, right? So now anything is possible in genetics. You can take this in agriculture or in antimicrobial resistance and so on and do fantastic things with it. And I could talk for 2 days on the potential applications, but you've got the point, and I'm happy to answer any questions you might have. Thank you for your attention.